Okay. So we have a few slides to finish up from last time, and then we have some new material. Real quick as we begin our time together, I just want to go through these key terms really quickly. We went through them in the video that I recorded, but real quickly we'll go through these again. So common disease, common variant. These are specific types of complex diseases that are common, that are due to very common mutations. And so they're very common mutations that tend to aggregate, and so every individual tends to start their life with several of these. And so this is one reason why complex diseases can be common, if the mutations that trigger this complex disease are very common. There are other common diseases that have very rare variants, but those variants, when they're present, have very large impacts. And every individual mutation might be rare, but if you only need one or two of them, and there are literally 1,000 different sites, you know, then it's like, well, yeah, any given one is very rare. It's just like my chances of being struck by lightning, or bitten by a shark, or eaten by a pig, or winning the lottery, or getting smacked in the face by somebody really famous, like all of those are very, very small probabilities, but the probability of any one of them happening, that's a little bit more likely, right? You don't need all of them to happen, and you don't need a specific one to happen. It could be any of them, right? You know, like I'll avoid pig farms and places where lightning strikes often, and swimming in the ocean. No, you don't want to avoid that one. That one's, you just swim in the ocean. The probability of being bitten by a shark is very low. All right, synthetic associations. So synthetic associations, basically the idea that sometimes these rare variants piggyback on more common variants. So you have very common mutations that tend to appear in, say, like an entire people group. Like, you know, some of these variants that we use to test ancestry. You know, like this is a very common mutation in, you know, people that are from the British Isles. And then some of these rare variants, population-wide, might be rare, but they might be more common in certain people groups because they tend to go along with more common variants. Okay, and so where the probability, you know, human-wide might be, you know, 1 in 1,000 or 1 in 10,000, it may be 1 in 100 in certain people groups. And even though it's still a very rare mutation, if it correlates significantly with a more common mutation, then in people groups with that more common mutation a lot might have this more often. Yeah?
So two quick questions. So when you say variant, you mean mutation?
Yes.
Synonymous here?
Yeah. Because the idea there would be that the only source of variation is mutations, but if your view of origins holds to that some of the variation is created and was put in place from the beginning, then variant doesn't have to be synonymous with mutation. But in our textbook, variant is synonymous with mutation because mutations are the only source of variation.
Okay.
Yeah.
And second question, how would it be able to piggyback on the more common variant? Like how would
it depends on where it's at. It'd be because they are not assorting independently. They're close enough together, and natural selection might be favoring a certain variant, and so this rare variant that causes some pathology piggybacks on it because they're not assorting independently. They're too close together on a chromosome, or they're too closely associated, even if they aren't close together, but the two portions of the chromosome tend to associate with one another. They're too closely associated with another location on the genome to assort fully independently. Stochastic fluctuations, these are random changes in how genes are expressed. Bonferroni correction, it's just a way to reduce your probability of making an error and making the specific error where you say they're different when they're really the same. Okay? And so you're like, it's a way of reducing that probability, but it makes the other type of error a lot more common, which is you saying they're the same when they're really different. Okay, and so Bonferroni correction, basically you just take your alpha level, which we usually set at 0.05, and you divide it by your total number of samples. So if you have 100,000 genes, you divide 0.05 by 100,000, and that's your new alpha level. That's the new place in which you say, okay, they're different. They're different. Thrifty gene, hypothesis. This is the, oh, you know what? We'll save all three of these because we're going to talk about them in more detail. Okay? Any questions about these key terms or thoughts?
Wait, just last thing, the
Bonferroni?
Yeah.
Yeah.
It makes the probability of error of saying they're different when they're actually the same?
Yeah, and I think it's called a type 1 error. Is that right? Those that have you taken statistics. So a type 1 error is basically we determine they're different when they're really the same. We determine two groups are different, but they are really the same. Now, when we're doing any kind of statistical test, we tend to set our alpha level at 0.05. And so if basically you're like, if there's a 5% chance or less of having your troops being this different, if they're really the same, then we're like, no, they're actually different because we have less than a 5% chance. But you're like, no, I mean, there's a 5% probability that they are actually the same, and you determined they were different, right? And so what we're like, if you're dealing with any one individual locus in the genome, and you're like, does this correlate with this genetic illness? And you're like, okay, we've got a 5% chance that we're going to be wrong. We're going to say it does correlate when it really doesn't. Okay, we're going to say like this specific mutation does correlate when it really doesn't. We've got about a 5% chance of making that mistake because our alpha level is 0.05. Okay, when we're like, okay, well, any one locus is not that big of a deal. We've got a 5% chance of making an error. If we're doing 100,000 of these, now you're like, we're going to make a ton of errors. 1 in every 20, based on that probability, if you've got a 5% chance of making a mistake, 1 in every 20 loci you're going to be wrong on. And you've got 100,000 of them. And 1 in 20 of 100,000 is a lot. What would that be? 1 in 20 would be 5, would be 50? 50, well, that would be 50 of 1,000. Yeah, 50 of them you're going to be wrong on. Okay, that's a lot. That's a lot. And so what we'll do then is we'll divide that 0.05. We'll say we're no longer going to use 0.05 as our cutoff. We're going to divide 0.05 by our total number of samples, in this case 1,000. And so 0.05 divided by 1,000 is now 0.0005. And we're like, that's the new cutoff. That's where you need to get to say, no, they're really different. Okay? To be able to say that they're different. You're like, okay, we've definitely limited the chances of making a type 1 error, saying they're different when they're really the same. But a type 2 error is we say they're the same, they are, so they're the same, when they are really different. So this is what we call a type 2 error. We say they're the same when they're really different. And so the chance of making a type 2 error is actually really high. And then when we make our alpha smaller and we make this less likely, we become less likely to say they're different if they're really the same. We become far more likely to say they're the same when they're really different. And for some things it's not really that big of a deal. But when you're talking about a specific mutation and whether or not it correlates with the genetic illness, and we have all of these that now we're very significantly likely to say they don't correlate when they really do. And we're like, wow, now that's actually meaningful. Like a type 2 error really matters there. And so that's an outcome of a Bonferroni correction. You limit your probability of making a type 1 error of saying they correlate, like a specific mutation correlates with the genetic illness when it really doesn't, but you make it a lot more likely to say it doesn't correlate when it really does.
What's the point of doing it then? Aren't both errors equally likely?
Well, I mean, it depends on which one's worse. And so is it worse to say that this gene does correlate with it and put a lot of effort and a lot of money into studying how, and it really doesn't correlate with the disease at all? Or is it worse to say that specific mutation doesn't correlate with it, and so you spend your money on ones that you know do, and then you maybe miss a few? And so a type 1 error costs more money and is probably a bigger issue than making a type 2 error, than missing some of the genes that actually do correlate with it. Because you still have some that you know correlate with it, or you're a lot more confident that they correlate with it, and so you can focus your efforts there and figure out how, what's going on developmentally. So here you'll miss some information. Here you would be putting effort into something that may just be completely wrong. So here you may miss some. Here you're basically wasting resources. Okay? And basically whatever costs more money, that's the bigger issue.
But what about like what costs more likelihood of like failing and more people dying?
Well, that'd still be the same here. Because here you're still going to have some mutations that you're really confident that they correlate with it. You might miss some, but you're going to get some that you're like, we're almost certain. I mean, there's no certainty in statistics, but we're almost certain that these mutations correlate significantly with this illness. And then so you still have something to focus on and something to use to predict that this person's going to be more likely to develop this genetic illness and work on prevention.
But if you miss one that's really important, that's also a big deal.
If it is really important, it shouldn't be an issue. Because if it's really important, your statistics should indicate that. Otherwise, the statistical tools you're using are useless. Okay. All right, so here are quantitative traits. And so something we mentioned last time is a quantitative trait is something that varies continuously. You've got a minimum value, you have a maximum value, and basically anything in between is possible. And so in the top left, it basically shows you the simplest kind of quantitative trait, in which you have a single locus, 2 alleles. Okay? Single locus, 2 allele. One allele would give you, let's just say this is height, right? This is really nothing, it's just giving you a mathematical example. But let's say it's height. And let's say human height was controlled by a single locus. You're like, how would that work? It doesn't matter. We're imagining here. Okay? Human height is controlled by a single locus. If you're double recessive, you're short. If you're double dominant, you're tall. If you're heterozygous, you're somewhere in between. Okay? And so we've got, okay, a single locus with 2 alleles determining a quantitative trait. And we're like, okay, even there, it's going to be really complicated to figure out how this all works, because if anything in between the short and the tall is possible with the heterozygous genotype, that heterozygous genotype is now obviously very heavily influenced by the environment, right? Because if it's like, if you're double recessive and you're short, you're double dominant and you're tall, and then if you're heterozygous, you're everything in between the short and tall. You're like, wow, so for double recessive, it's pretty simple. Double dominant, it's pretty simple. Heterozygous, this is complicated. It's complicated to be able to make a prediction on what that person's phenotype is going to be. The reality is no quantitative trait is really like this. Quantitative traits are like this, where you've got dozens of different loci, each with dozens or hundreds of different alleles. And so we've got a really nice continuum and a nice bell-shaped curve. For our quantitative traits, which again have a continuous distribution, you have a minimum value and maximum value, everything in between is possible, you tend to have most of your individuals having whatever that average condition is. Okay? And so the problem with this, the problem with this is it becomes very, very difficult to be able to predict from the genotype what the phenotype is going to be. For several reasons. One is you have multiple different loci impacting the phenotype. Okay? And so it's hard to figure out, okay, well, which gene is most important? Which gene is least important? Which combinations are particularly helpful if you want to be tall? Which combinations are particularly helpful if you want to be short? Right? And you're like, why would you want to be short? Well, if you want to be a jockey, you've got to be small. Right? You've got to be under 120 pounds, I think. You're not going to be 5 foot. You're not going to be 5'11" and be under 120 pounds. I mean, if you are, that's going to be weird. And you're probably not going to be strong enough to ride a horse, anyways. And so it's really complicated just because there's so many different loci and so many different alleles complicating this. But what makes it really, really difficult is that if you're really getting something like this with the phenotypes, it's an indication that environment is playing a really big role. That it's not just genotype. That you can go from the genotype and make predictions, but the environment's playing a heavy role as well. Because what you're going to find when you have a nice bell-shaped distribution like this, and you go and look at a bunch of people at the average condition, guess what? They're going to have completely different genotypes, right? Most humans are pretty close to the average height. An average height for a male in the United States, I think, is 5'9" or 5'10". 40% of the population is within one standard deviation of that. And yet, super different genetically, right? I'm 5'11". Who else in here is 5'11"? Anyone? You're not 5'11". Nobody else in here is 5'11"? Oh man.
5'10" and a half.
5'10" and a half, just perfect. I'm actually 5'10" and a half as well, but I round up because 5'11" sounds better. And so, but Christian and I, if you went and look at our genomes, we're not going to be identical genetically, right? Christian and I are not identical twins. And even if we were, you already know identical twins aren't even really identical genetically, right? And so that's the problem, is because environment plays a huge role in all of these quantitative traits. And so they become really complicated. So whenever you see this with a phenotype, you're like, man, if it really matters, we have no idea how to make things work, right? You can take two tall people, and you're like, you're going to have tall kids, and then they don't. They have short kids, and you're like, what on earth happened? You're like, I don't even know. I fed them milk. Like, I don't get it. Alyssa.
Could it also be, though, it's not just, environment does play a role, but even if, say, environment did play a role, you can still have that bell-shaped curve, because there's so many different things that, contribute to it. So one person might have something that contributes to them being tall and being short.
Sure.
So everyone has a different combination. And then
yeah, but it doesn't happen.
No?
No.
That's not how
Is it theoretically possible? Sure. But it doesn't happen. Whenever you have multiple genes controlling the same phenotype, environment's always going to play a huge role because both epistatic and epigenetic effects are going to be huge, right? And so if epigenetic effects are huge, environment determines a lot of your epigenetics. It's determined by your environment, and it's just like, is it theoretically possible? Yes, but it's not going to happen. It's not going to happen. And so you're like, well, human height is not really that big of a deal. Who cares, right? Who cares? And you're like, well, people care. But what if this is like the severity of a genetic illness? Right? And so what we're mapping on this is a particular genetic illness, and the average condition is basically like, you're probably going to get to, I don't know, maybe 60 before you start to experience pathology here. But, getting towards the extreme, it's like you're going to start to experience pathology in your preteen years. And then here it's like you're never going to experience this pathology. And we're like, well, now this continuum is really important. Right? And the average condition is you're probably going to get to late in life, and then you're going to start to experience some pathology associated with this. And now it's like, well, now this really matters. But we know when we're going to have a nice bell-shaped distribution like this with a phenotype, man, we know that environment's playing a huge role, and we know that there are going to be several loci, and it's just going to be really complicated. And it's like we can predict and try to prevent, like, hey, you're more likely to experience pathology, so you might want to try doing, these things early in life. But other than that, it's just like, we just don't know. It's not as simple, right? Unfortunately, not everything is like pea plants, where pea plant height is determined by a single locus, and they're short and tall, and there's nothing in between. Yeah, John.
So two tall people having a child, you can't just say, oh, that kid's gonna be tall.
No, you can talk about probabilities. You can talk about probabilities. Like, so if two tall people have a child, their child's significantly more likely to be tall than two short people. But it's like it's not as simple.
So many loci.
Yes.
Based on environment.
Yep. And then you don't know why they're tall, right? Some people are tall because they have like some kind of rare genetic illness, like Lincoln did. He's all gangly and tall. It's because he had a really rare genetic illness. You didn't know this? Oh, I think they think he had acromegaly. But anyways, I don't know. I don't remember. It just came to me, and then I shared it, and I don't even know if it's true. I didn't prepare for this class by reading about Lincoln.
So then it sounds like, so we have a general idea of, okay, so we know that environment affects the loci, et cetera, but we don't know specifically how. So we have a general
Correct.
idea, but we don't know the specifics
Yep.
of what affects.
Now we're getting better. I mean, up until about 10 years ago, nobody really did anything with epigenetics. And now we're getting really good at mapping epigenetics and even giving an epigenetic sequence and a code. So we're getting better, but yeah, I mean, we still know very little. Yeah. First law of technology, right? We overestimate the initial impact and underestimate long-term impact of any new technology. All right. So here are some of the issues with quantitative traits. This table comes right out of the text. It doesn't look amazing on the slide, but it's okay because you have the text. Genetic heterogeneity, and again, this is the issue where there are several different ways to get to the same phenotype. Several different ways to get to the same phenotype. And this is especially true with these complex diseases. There are several different ways to develop type 2 diabetes. Several different ways to develop various types of cancers. Several different ways to develop these particular phenotypes, and so it's really complicated. It's like, you know, and several different ways to develop a height of 5'10" and a half, right? Several different ways. It's complicated. It's messy. Phenocopy, and so this is basically that you can end up with a phenotypic condition and have none of the genetic signatures that are associated with that. And that's because the environment can trigger that development, right? You can be really short and have all of the programming to be tall. It's just you had a really poor diet as you were developing, right? Or your mom was struggling to keep you nourished when you were in utero or nursing. And it's like, man, sorry, like you were supposed to be tall, but it didn't work. It didn't happen. It didn't happen. And then pleiotropy, and this is basically where you have a single gene that impacts several different phenotypes. Okay, and so there are some genes that it's where, you know, the two people could have the same exact genome, which we know isn't even true for identical twins, but let's assume it were true even for identical twins. Same exact genome, and yet they could be different phenotypically. And it's like, well, that could be environmental, but it could also be these, what we call these stochastic effects, where you get kind of a random expression of various genes. Incomplete penetrance is something we talked about a long time ago, that individuals could have a genotype for a particular phenotype, but it just not come because the penetrance is less than 100%. And then the exact diagnosis is difficult, and this is just because, again, symptoms change throughout time, and then the same symptoms might be associated with several different illnesses. So now you're adding complexity here. The same illness may be caused by several different genetic mutations, and then several different diseases can have the same symptoms.
Isn't the last point there kind of the same as the first point?
The first point? Well, I mean, this first point you would make for like a specific genetic illness, a specific character, and then this is like symptoms would be like a physiological impact of a character.
Oh, so it's the opposite.
No, it's not the opposite. They are certainly related. Like if you've got, let's use abdominal pain. Abdominal pain is one of, abdominal pain, headaches, right? These things that there's several different causes of these, right? And so you could have abdominal pain because your appendix is about to blow up, right? Or you could have abdominal pain because you're lactose intolerant and you ate an entire block of cheese, right? Why? Because maybe you didn't know you were lactose intolerant or you just didn't care. Cheese is delicious, right? And so, yeah, this adds some complexity to this. This we're usually talking about like a very specific genetic illness, several different ways in which you can get that genetically. And this is like, we may not even have the disease right? We're thinking this person was diagnosed with this disease, and we're trying to figure out what in their genome maybe triggered this or made them more susceptible to it, and our diagnosis is completely wrong. Right? Wow, that thing sounds really angry. So this one's kind of fun. So this is like, okay, so we do these heritability studies, and we basically, we figure out that, human height, 80% of the variation in human height is due to genetic differences. But then we go and we try to do genome-wide association studies, and we find every loci in, or I guess it'd be every locus, yes, because you have to use singular. Every locus in the human genome that impacts human height. And we basically study each one individually and figure out how much of the variation in human height is the result of variation in this locus. Okay? And we're like, cool. And then we go to the next one. How much of the variation in human height is because of this locus? And we're like, cool. And then we go to the next one. We're like, how much of the variation in human height is because of this locus? And we're like, cool. And then we do that for all of them. And we add up all of that variation. And we're like, we've only explained 5% of the variation in human height. And we're like, what's going on here? We know the heritability of height is 80%. So 80% of the variation is because of genetic differences. And yet, we're only explaining 5% of that, and we have all this, what we call missing heritability. Missing heritability. And we're like, what does this even mean? What does this mean here? It's supposed to be 80%, but we're only explaining 5% of that. Either, one, we're missing additional loci, although the probability of those doing anything is pretty low, right? Because if we've already got several hundred and we're really confident that those, and then we find some more and we're like, this might impact human height, and we're like, okay, we'll start adding those in. It's really not going to significantly change our explanation. So either we're missing some data or our methods are completely wrong. And it's like, okay, missing data, that may explain some additional variation, but it's not going to get us from 5% to 80%. So our methods are completely wrong. And you all know, as well as I do, what the issue is of taking one locus at a time, figuring out how much it impacts the variation in height, and then adding those up all together at the end. What's the issue then? What's that?
You don't see how they work together?
You don't see how they work together. Nothing in the genome works in isolation, right? It's not like you have all these different parts of the genome and they're all building their little products, and then they're like, hey, sorry, you gotta wait until like my contribution to human height is done. Like your contribution's over, you know, we're just gonna kind of relax and then I'm gonna come in. That's not how it works. Like the genome, everything is connected. Everything's related. You know, chromosome 1 sometimes interacts with a portion on chromosome 4. Portion on chromosome 5 might interact with a portion on chromosome 8 to control gene expression. And then we've got all these epistatic effects, right? Where the expression of one gene influences the expression of another. And we're like, wow, it's a really bad way of doing this. And then so what we do then is we're like, okay, we'll develop better statistical tools and fill in some of this missing heritability. And we'll start looking at all of the, you know, variation at once instead of one at a time. And it makes it complicated, and it makes it to where, you know, I mean, doing one at a time, after taking a college level statistics class, you could sit down and do that analysis. Like I could give you 100 loci, I can have you figure out like what percentage of the variation in human height is due to each of those, and then you can add them all up together at the end. You could do that with a single college level statistics class. You can't look at the effects of all of them working together and influencing each other with a college level statistics class. I mean, we're talking about very complicated and heavy level of analyses. But there are people that can do that. And so finally biologists are like, you know what, we're not that good at statistics. We're just not that good at statistics. We're okay at it, right? We can publish, you know, something in our findings and do some of our own analyses, but we're really not that good at it. So why don't we use some statisticians that are so good at statistics that businesses pay them enormous amounts of money to predict market trends? It's like, well, they're probably pretty good at statistics. Let's start using them. And so this is what molecular biologists have started to do. They've started to use these statisticians, and we're getting a lot better. And so now I think the most recent publication, we know that the heritability of human height is 80% from doing studies within relatives and close relatives, and we can explain about 70% of it. About 70% of the variation we can explain. So we're not missing all that much now. And what we're missing is probably now missing data and not issues with the analyses. All right? But now it's like the unfortunate thing about doing that and employing statisticians is now when you read the papers you can't understand them. I mean the methods. Unless you're a statistician, you're just like, okay, I guess I'll take your word for it. I understand your analyses, but I don't understand your actual test. All right. So this question is a good one. Why doesn't the genome predict everything? Why doesn't the genome predict everything? Why can't we sequence our genome, which you could do so for about $1,500, at least sequence 96.5% of your genome, for about $1,500, and why can't I just look at your genome and predict everything that's going to happen to you in your life? Right? They did it in Gattaca, although they were wrong there too, because, spoiler alert, guy totally didn't care what they predicted at his birth, right? It's like, I'm going to become the man I want to be, and then he gets on a rocket ship and goes to Titan. So, spoiler alert, the movie's 20 years old, you know. Is it 20? I think it is 20 years old. Anyways, Gattaca. It's a pretty good movie. I mean, I cannot suggest that you watch it. So this is in no way a suggestion that you should watch the film or an endorsement of anything stated in the film. Okay, so why doesn't the genome predict everything? First of all, we've got epigenetic changes. And so we know this from our work on previous classes about epigenetics. We can mask the effects of a mutation. We can completely mask a single nucleotide polymorphism, right? You could have a super rare single nucleotide polymorphism that would be devastating if it were expressed. But if you methylate that region, it doesn't matter. It doesn't get expressed, right? We can mask it. We can also accentuate the effect of a variant, right? It could be a very minor change, but we overexpress that region of the genome, and now it's like, well, now this little change is now a big effect, right? And epigenetic changes can do this. And so if you want to really accentuate something, you acetylate the histones, right? And then so it's going to really increase the amount of transcription there, or the amount of activity there, and it might be a very minor issue, but now we've made it big, right? We've made it big. Or you can completely mask it if you methylate the histones or methylate the sequence directly. Alyssa.
Well, I was going to ask if this is on the PowerPoint, but I think it's on different.
Yes, you're still looking at the one from Monday. You got to get the slides I put on from Wednesday. They're on Canvas now. And then, so something we have to consider with epigenetics is these features are different from cell to cell, right? Every different cell type in our body has different epigenetic signatures, which is how you get various degrees of expression and how you get division of labor, right? How do you keep liver cells from making proteins that only immune cells make? You shut down those genes in liver cells. And how do you do that? You methylate the promoter, right? Or methylate the histones around that. Okay, so the epigenetic features differ from cell to cell. They change throughout the life of a cell. Your diet, your particular environmental stressors. I mean, the amount of times you're hugged as a child, right? All of this is going to impact epigenetic signatures in your cells. And then they are also going to vary from generation to generation. As you imprint your genes differently than the ones you inherited, because the genes that you have with your mom's imprinting, when you pass those on to your gametes, you're going to put the male imprint on them if you're male. And if you're female, the genes you got with your father's imprinting, when you pass those on to your gametes, you're going to put maternal imprinting on them, right? And so these epigenetic features change from generation to generation. And this is complicated. We're just figuring this stuff out, right? We've been studying DNA for 70 years. 70 years. I don't know why I looked at my watch. For 70 years, we've been studying DNA. I mean, really, if you want to go back to Mendel, and you like Mendel studying inheritance, he knew nothing about DNA, but did some pretty good work on inheritance. Of course, nobody really discovered Mendel's work until like the 19-teens and 1920s. But anyways, so we've only been studying DNA for 70 years, but we've done a lot. We've done a lot in 70 years. We've been studying epigenetics for about 10 years. Okay? We're just getting started. We're just getting started. We've literally been experimenting with self-driving cars longer than we've known about the importance of epigenetics. Wow. And we still don't do a very good job with self-driving cars. Right? They did a self-driving trolley that hit the streets of Vegas about a year ago with no passengers in it, and it made it, I think, 8 minutes. 8 minutes, and then it ran into a wall and, self-destructed. And we're like, cool, let's start putting people on self-driving trolleys. Or let's start, self-driving big rigs, right? We want people to be able to stay home with their family and not spend 3 weeks on the road. Let's just put out self-driving big rigs. Of course, if you hack into them and you shut down all the freeways leaving a city, you got a problem. But anyways, first world problems. Okay. All right, and then the other one, epistasis. So studying individual SNPs, single nucleotide polymorphisms, really doesn't tell us much about heritability. And it's because it's completely silly. It's completely silly. Be like if you want to study how a society works, and you're just like, you know what, I'm just going to study all of the people as individuals. It doesn't make any sense. Society isn't built up of individuals that don't interact, right? The whole point of society is you've got interactions and it increases complexity. Right? It just doesn't make any sense. And so we know that the expression of one region influences the expression of another. We call this epistasis. We know that the expression of one region of the genome can influence the expression of another. We know this. And so we're like, why on earth would we ever have thought we could just look at all of these individual SNPs and then aggregate the effects and say, we got it. We got it figured out. We got it figured out. That's not how things work. And so these epistatic effects, what they'll often do is combine the effects of different variants. They'll combine the effects of different variants. Yeah.
So, but could you study them individually? If you took out a certain gene, a random gene, and then you figured out studied the effects of it one by one, that could help because then you're figuring out how it affects the rest of the genome.
Maybe it would help only if the expression of both genes wasn't related in any way. So, like, let's say an individual gene just by itself. So you knocked out all other genes, right? And you're just going to study, okay, how does this gene influence height? Okay, and so you knock out all the other genes and the individual still survives, even though they wouldn't, but let's assume that they survive. And you're like, okay, this gene, if you have this particular variant, it will increase height by 5%. You're like, cool. Then we're going to knock out that gene, we're going to bring back one of the ones we knocked out previously, and we're like, all right, well, and then we're going to be like, okay, this specific mutation in this gene increases height by 2%. And you're like, okay, so from that, your idea is, if you have this specific mutation at that one that increases 5%, and this specific mutation at the other that increases 2%, if you've got both those mutations, you're going to have an increase in height of 7%, right? 5% from the one, 2% from the other. But what you find is, when those two are in place, it'd be more like 10% or 15%, because the effects of those compound. Because what you find is that a lot of these genes that are associated and are influencing it aren't actually coding for a single protein. They're coding for dozens of different proteins. And not only that, some of those proteins might be transcription factors that increase the transcription of other sites.
But I mean, couldn't you find that out by just knocking it out? So now instead of just studying one, you get rid of it in the genome and see that it goes down by 10%. You're like, oh, okay, so.
Right, but you don't know if it went down by 10% because you were missing that specific mutation, or if it went down by 10% because you were missing that mutation and what it did to a second mutation. So it's not that simple. It's not as simple as you want me to believe it is. I know. Listen, I'm with you. Like, I want this to be really simple. I want to be able to sequence my genome, and I want to be able to tell on what day that I'm gonna die. I want to be able to do, actually I don't want to do that, but that'd be kind of fun. Like, to know like if I knew exactly when I was going to die, it wouldn't change the way I live my life, but it'd be cool. Like, I'd probably wear like a clock, and so like it was counting down to that, because it'd be fun. Probably wouldn't change the way I live my life at all, because I mean, doctors tell me all the time, like, hey, if you don't stop doing this, like, you're probably gonna die. I'm like, I like it. We're all gonna die, doctor.
Heaven, baby. Heaven bound.
Yeah. Like, I mean, I had to have a heart-to-heart with my primary care physician one day. I'm just like, listen, you're gonna die too. Being a doctor is probably not great for your health. It's a stressful job. Do you consider doing something different? I have a job that has no stress, so I mean, how do we factor that in? So. You guys don't talk to your primary care physicians like this? I have Kaiser, so I don't have a set primary care physician that I have to go to every time. Every time I get a different one, right? And it's like, how many of these can I go through before I'm like, I have to go to a different office? It's like, we don't talk to you anymore.
So is it the idea of, let's say you have an engine in a car, you want to sit there and watch it as a whole interacting?
Oh, absolutely. That's a great analogy. Yeah, that's a great analogy. It's like, yeah, I mean, an engine is far more than just the sum of its parts, right? And just the genome is far more than the sum of its parts, right? One piece influences another piece, and we call these epistatic effects. Now, in pure epistasis, we're talking about the expression of one masking the effects of another. But really, anything in which the expression of one gene influences the expression of another, that's an epistatic effect. It's an epistatic effect, and it complicates it. And the thing is like, it's somewhat random. And this is, in the key terms, we had this stochastic effects, right? Where it's somewhat random. Right? Sometimes these two genes, when they work together, will give you a 15% increase. Sometimes they'll give you a 12. Sometimes they'll give you a 7. And it's like it's somewhat random, because really the idea there is it depends on which one starts getting expressed first. Which one starts getting expressed first. You know? Like if my brother and I teamed up to do something, and I got to go first and do all of the hard things, and then he went and just finished whatever was easy, we would be a great team. But if he went first and tried to do all the hard things first and screwed them up and didn't get anything done, and I had to come in and try to do everything, we wouldn't be that great of a team. That's actually a horrible illustration, because my brother and I would probably be the flip. My brother's really good at everything. And he's so tall and handsome, you know, and he still has a full head of hair. He's like 3 inches taller than I am. And he does have a full head of hair. And his beard is way better than mine. But, you know, what are you going to do?
You're smarter though.
I don't think so. Yeah, I definitely am more educated. He only has a master's degree, so. All right. Why are the complex diseases more frequent today? This is what the rest of chapter 9 is dealing with. Why are we seeing an increase in these complex genetic illnesses? And there are several hypotheses. One, this was my favorite. I talked about this in the key terms, the Thrifty Hypothesis. And it's basically like we had certain mutations, certain variants in genes that made it really helpful for us to put on fat easily and store up some resources for periods of famine, for periods periods of famine when you didn't have enough to go around. And we're like, yeah, it works really well when you may eat one day and not eat for 3 or 4 days. But it doesn't really work all that well when you're eating 3 pretty nice meals a day. And so I like this one, the thrifty gene hypothesis, right? We have genes, or we have mutations in those genes that were selected by natural selection or favored because they helped us survive periods of starvation, and we're not starving now. We're not having periods of starvation unless you do intermittent fasting. You're like, I'm going to fast for 72 hours because that's the way humans were designed to live, right? Eat a big meal and then fast for, 72 hours. The hygiene hypothesis. So this has to do with like autoimmune diseases, various other things associated with issues with your immune system, maybe allergies, autoimmune diseases, immunodeficiencies, and it's the hygiene hypothesis. And this is basically the idea that you need to give your immune system something to fight. Your immune system needs to be exposed to foreign material and needs to learn how to fight certain pathogens. It needs to learn how to fight bacteria and viruses and parasites. And when you live in a really clean society where your water is super purified and your food is super purified, and when you try to stick your nasty, muddy hands in your mouth, people are like, no, don't do that. It's like you don't give your immune system something to fight. I totally let my kids do that. I believe in the hygiene hypothesis. I'm like, eat that mud. Just eat it. You want to make sure your immune system matures properly. And so this is also referred to as the Th One Trigger Defect. And so what we mean by there is you have 2 different types of T helper cells. You have T helper 1 cells and you have T helper 2 cells. T helper 1 cells trigger basically the response from antibodies. Th1 cells, they get the cells that make antibodies, B cells, and the other types of T cells that make T cell receptors, they get those to respond to things like bacteria and viruses. Th2 cells, these are Th1. So Th1 cells get B cells and other types of T cells to really start to accumulate. And these are for fighting like bacteria and viruses and small parasites, intracellular stuff. Th2, these get the cells that are associated with really big pathogens to work, like macrophages and eosinophils. These things that they're not making antibodies, they're not making T cell receptors, they're attacking really big things. Like you've got a parasitic worm living in one of your vessels, and you're not going to just coat it with antibodies. Like, it's just going to shake them off, right? Well, then it's going to shed its outer covering, and the antibodies are gone anyways. Like, you've got to get cells to start chewing bites out of that parasitic worm. And so what you find is at birth, you basically are programmed with the Th2 response. And you have to be exposed to bacteria and viruses in order to start to develop the Th1 portion of your immune system. Otherwise, it doesn't develop properly, it doesn't mature properly, and you start making antibodies against everything. Making antibodies against, pollen and pet dander and ketchup and your own proteins. And that's where you really got an issue, when you start making antibodies against your own tissue. Okay? So you need to mature this aspect of your immune system, the Th1 portion, where you give these cells that make antibodies something worthy to fight. Yeah?
So you think people who aren't exposed to anything like this, little babies, will be a little more likely to develop allergies and stuff?
It is one explanation for why allergies and autoimmune diseases are so common in developed countries. And yes, I think it's true. Like, your immune system needs to learn how to fight proper pathogens. Yeah?
Is it possible that by being surrounded by a bunch of different things that you're actually causing your immune system to become overactive?
Oh, sure.
Itself?
Yeah. Now then the question is, well, what's worse? Is it worse to have an increase in infant mortality or to have an increase in immature immune systems? And it's worse to have an increase in infant mortality, right? So it's like, we've got a greater chance of surviving to reproductive age. That's good. And this is just one of the implications of that is your immune system's probably not going to mature properly.
Or we could just explode a little bit.
You could. Like maybe give somebody a vaccine or something? David.
There's some studies that children that live on farms have higher immunity.
Yeah.
Basically, people that live with animals too, that
Yep.
dies and improve the chances.
Yeah, and it's not being exposed to animal antigens, it's being exposed to all of the bacteria by interacting with your dogs, right? And being exposed to parasites. Yeah, I mean, it's just, there's a lot of evidence in support of the hygiene hypothesis. So the old friends hypothesis, this is a subset of this, and it's not like, the old friends hypothesis is basically this. The hygiene hypothesis would just suggest hey, you need to be exposed to bacteria and viruses in order for your immune system to mature properly. The old friends hypothesis says, no, you need to be exposed to specific bacteria and viruses. The things that our species has always interacted with. And the idea here is our immune system basically has evolved throughout human history, and this is really regardless of your view of origins, to fight specific pathogens. And you need those specific pathogens. And we're trying to figure out what these are so that you could do that, Alyssa. You could give your kids just a little bit of it, right? Sprinkle it on their food because a little additive. So, we're out of time. There are a few more hypotheses on these that you'll need to read over before you can address the quiz, but you should. We haven't talked about all of them, and one question on the quiz does deal with these hypotheses, so you'll need to kind of look over the remaining, but they're very straightforward. These are the big ones, and then the other ones are more minor explanations.
Where do you look for them?
On the slide.